For 50 years, the Nobel Conference at Gustavus Adolphus College has hosted preeminent scientists, theologians, and ethicists to discuss deep questions in the intersection of science and society. From the newest results in physics, chemistry, and biology, to the newest fields of multidisciplinary study, scientists at the Nobel Conference have examined the universe at its largest and smallest scales, explored the oceans, and described new materials. Conference speakers have debated the mechanisms of aging, as well as science and economics of food. Often, speakers have given us a glimpse of the next big questions and how they might be answered. Throughout all of the conversations, ethicists and theologians have grounded the science in a human dimension. The 50th Nobel Conference at Gustavus is celebrating a half-century of bringing breakthrough science to lay audiences in the upper Midwest, across the country, and recently around the world. Nobel Conference 50 is assembling previous Nobel Conference participants to look at recent advances and future directions in the physical sciences, evolutionary biology and ecology, medicine and physiology, and the intersection of science and public policy. Sean B. Carroll is the Alan Wilson Professor of Molecular Biology and Genetics at the University of Wisconsin-Madison, where he has built a research career focused on evo-devo, the genetics and molecular biology of animal development, and how that relates to the evolution of morphological diversity. Dr. Carroll leads the Howard Hughes Medical Institute's Department of Science Education, the largest private supporter of science education activities in the U.S. We hope you enjoy this year's presentation by Sean B. Carroll, entitled Evolution at the Molecular and Planetary Scale, A Tale of Two Biologies.
Thanks, Colleen, and thanks to Gustavus for having me back to this extraordinary event. I try to tell my colleagues when I'm going off to do this trip what this is like, and there's just no description unless you've been here. This is a unique event in the scientific and educational world, not only just in the US and around the world. So thanks for having me back, and thanks for being so, I guess, liberal-minded, because I'm not a Nobel laureate. I'm not Swedish. And I'm from Wisconsin. So what I want to do today is, I tried to picture, you talk about 50 years, and it turns out just on so many scientific fields, the last 50 years have been so momentous. I thought, what would I have talked about 50 years ago if I was here? I was 4 years old. I'd have talked about green eggs and ham and Captain Kangaroo. But we're going to do a little bit of looking back and a little bit of looking forward. And sort of the theme of this is we're going to look both at the molecular scale and the planetary scale, and I call it a tale of two biologies because I'm just ripping off Dickens. And really what I want to talk about is how, really for evolutionary science, the last 50 years has been the best of times. But for evolution on the planet, well, you know the rest. So we're going to get to that a little bit later. And I want to look ahead and sort of ponder what might people be saying 50 years from now about the science that's going to transpire over the next 5 decades. So it's been the best of times for evolutionary science, really because of some pivotal discoveries. And the discovery that really ushered in a new golden era was the discovery of the structure of DNA in 1953, which was awarded the Nobel Prize in 1962. And the reason why this was so important, the solving of the structure of this molecule, is that really the structure had to solve two huge mysteries about life. It had to solve the question of the stability of life, how traits are faithfully inherited from generation to generation, why dogs have dogs and cats have cats and people have people. But it also had to explain the mutability of life, how mutation generates variation that enables life to evolve over time, how we wound up with cats and dogs and people and pigs and fruit flies. And the solution of the structure of DNA was not just an inflection point in modern biology, because it led to all sorts of things. In fact, I would say it pretty much now dominates the pharmaceutical industry, has a huge impact on agriculture, etc. But it was also a profound new insight into the nature of life. And Nobel Prizes are recognize a variety of things. Sometimes it's a really important technological innovation, and sometimes it's just a new profound perspective on our place in the world. And one of the co-founders of molecular biology, and one of the important biologists to me, Jacques Monod, was a 1965 Nobel Prize winner in physiology or medicine. He thought that all these technological things that science brings they're fine, but he thought that the most important results of science have been to change the relationship of man to the universe, or the way we see ourselves in the universe. And I really subscribe to this viewpoint. And there are two really huge ideas that biology has put forth that have revolutionized our perspective of ourselves and our place in the world. And the first, starting with the voyages of Darwin and Wallace, that led us to the realization that the origin of species is natural, not divine. And the second, really from the revolution in molecular biology a century later, was of the overwhelming evidence for the role of chance in shaping the course of life on Earth. And it was Monod who really put this together in a book that he wrote a little bit after getting his Nobel Prize. And he said in Chance and Necessity that chance alone is at the source of every innovation, of all creation in the biosphere. This central concept of modern biology is no longer one among other possible hypotheses. It is the only one that squares with observed and tested fact, and nothing warrants the hope that on this score, our position is likely ever to be revised. Now, interestingly, he said this where, from my yardstick, there was very little empirical support for this assertion. Yes, we knew something about the structure of DNA, and yes, we inferred something about how it's copied and how mutations happen. But I would say we didn't know a lot about the generation of diversity in the biosphere. Flash forward 50 years, and it's been a remarkable run. Today, and I'm sure high school students as well as college students can now interrogate the DNA of any creature that they want. We can determine the entire sequence of the billions of bases of DNA of any species or of any individual in this room. We can mine that DNA for the record of evolution to see how these chance mutations arise, very precisely, and how they generate diversity. So it's a new golden age of evolutionary science, enabled by our power to peer into the DNA record of any creature and really understand how that creature came to be, to see sort of how the fittest are made. So I want to do for a few minutes is just give you a few stories out of that DNA record, a few creatures that I think you might enjoy, that I think a few insights from their DNA that might stick with you as you think about your own DNA and your own history to this point on planet Earth. So let's start with what is, for me, the most remarkable creature I know about on the planet. It's this guy. This is known as an icefish. This is footage of an icefish swimming along the bottom of the Southern Ocean near Antarctica. You can see it's a little funny-looking fish, pretty light-colored, almost transparent. It's got a big mouth. He's actually a carnivore. Now, what's remarkable about this fish are many things. First, where it lives. It lives in water that's about 29 degrees Fahrenheit. And when you think about that in terms of physiology, the first question is, how is it that this thing doesn't freeze solid? All right? It's in really cold water. There's lots of little ice crystals floating around in that water. And if that fish, for example, in grabbing prey, were to ingest those ice crystals, well, those ice crystals can nucleate the formation of ice from the inside out, and you've got a fish stick, okay? But biologists many decades ago, peering into the blood of the icefish, found out that it and some of its close relatives has come up with a pretty nifty invention, one of those innovations that Monod was talking about. Its blood is full of antifreeze, a specialized group of proteins that prevents the growth of ice crystals, both inside the tissues as well as inside the blood. But what's really remarkable is that we can then look inside the DNA of the icefish for the code of the proteins that do the antifreeze job. And by staring at that code, we understand that the antifreeze came from another gene, a gene that was involved in digestion. And a little piece of the code broke off of that gene and was tinkered with a little bit by the process of mutation. And what resulted was the antifreeze that has now enabled fish bearing this antifreeze to dominate the habitat of the Southern Ocean, to live where other fish couldn't. Sharks, skates, etc., these things are gone, long extinct in the Southern Ocean as the water's chilled there over the last 30 million years, and antifreeze-bearing fish predominate. Now, the story of evolution is, of course, not just about gains. There's other sorts of changes that take place in DNA, and the icefish presents us with a really stunning example of that. If you were to open up this icefish or draw a little bit of its blood and compare it with virtually any other fish on the planet, this is the shock that you'd find. On the right-hand side here is the icefish blood, just this white, sort of pretty fairly dilute blood, compared on the left with the blood of a typical red-blooded fish in the Southern Ocean, a rock cod. What is going on here? When naturalists first discovered the icefish and drew its blood, they were completely shocked because this is the only fish, it and about 10 other species, that has, of all backboned animals on the planet, that have no red blood cells. So the course of life on this planet for 500 million years, there have been backboned animals with red blood cells in their blood carrying oxygen around to their tissues, but not in the icefish. What is going on? Well, think about that habitat that it's in. So yes, they're the only vertebrates without red blood cells, and if you were to centrifuge this tube of blood, spin it in a device that would then concentrate the cells, the only cells you'd find would be white, and they'd only be about 1% of the total volume of blood. Now, if you do that for typical fish like the rock cod, it's about 15 to 20% red blood cells. If you do that with yours or my blood, it's about 45% red blood cells. I mean, our blood is packed with cells carrying oxygen. And we know that if that number in you and I drops much below 45%, we call that anemia. And if it drops much lower, that's life-threatening. So how can an animal get by with no red blood cells? Well, in the Southern Ocean, it's so cold that moving fluid through a tube is kind of tricky. It's kind of viscous. So it turns out that the icefish, instead of sequestering oxygen in its cells, it just passively absorbs the oxygen from the surrounding seawater. The fish has no scales. It has enlarged capillaries up by the surface. And so really, the oxygen is almost in equilibrium with the surrounding seawater. So it has no active mechanism for transporting oxygen whatsoever. But that keeps its capillaries clear. Its blood is only 1% cells by volume, so it's really sort of dilute, quite dilute blood. And it has an enlarged heart, so it just pumps more of this dilute blood around, and that's how it's adapted to living in such cold water. Now, pretty extraordinary to get away without red blood cells whatsoever. And we might ask, what's happened to this icefish? And if we look in its DNA, there's a pretty dramatic story there. So if we look in the DNA of, say, a red-blooded fish, and if you look at the top of the slide here, I'm just showing you schematically that you can see the code for two genes that encode the different parts of the hemoglobin molecule that carries oxygen and you and I and everything else with a backbone on this planet. If you look at the corresponding part of the DNA in the icefish, what I'm showing you here below, only part of one of those genes is present, this red striped gene here on the left, and the green one is completely missing. And if you look at the code of this red striped gene, it's really eroded. It looks like a heavily redacted CIA document where lots and lots of the code is missing and it's mutated to gibberish. So essentially what you're looking at is a molecular fossil in the DNA of this animal. It tells us that the ancestors of icefish had red blood and had globin genes, but now there's no functional globin in these fish, and there's just a little remnant in its DNA of the lifestyle of its ancestors. So really, the DNA record of icefish and of all species reveals what was gained and lost and modified in the course of evolution. And when we look at particular genes, the shifts in lifestyle, I'm trying to move my arrow here a little bit, sorry, I'm a novice at this. Goodbye, arrow. The shifts in lifestyle that have happened as animals invade other new habitats, as the world changes, as climate changes, These are reflected in the DNA of pertinent genes. Some genes stay the same for long periods of time. Some genes take on new jobs, like antifreeze. Some genes disappear and no longer function. So living in the cold is an innovation, and we see the record of that in the DNA of this fish. Let me give you another example, one of my favorites, about an innovation and how simple these things can be in their origin. I'm going to tell you a story about a little hide-and-seek game going on in Northern Europe. This is a not-to-scale picture of a vole, a European vole. It's kind of a Monty Python-sized vole, so you could see it. They're so small that if I actually hid it in the grass here, you wouldn't be able to see it. Well, the animal that wants to find these voles is the European kestrel. And I want you to put yourself sort of in the shoes or talons of this kestrel and think there you are flying over the fields of Northern Europe, and you're looking for your lunch. How are you going to find this little animal in that thick green foliage? All right, so how does the kestrel win at this game of hide and seek? All right, so to explain that, I got to tell you a little bit about how we and kestrels and other animals see the world. And you all know about sort of the spectrum of visible light, what we would see as the rainbow. And that is the different wavelengths of light that we see that are coming from the sun, from blue through the orange and reds. But you also know that at one end of the wavelength there's infrared, at the other end of the wavelengths there's ultraviolet. Well, we see this, what we call the visible spectrum, out of our chauvinism. We call this the visible spectrum. And birds and old world monkeys and apes can see essentially the same spectrum we can, because they have the same kinds of proteins in their retina as we do that are excited by different wavelengths of light. And that excitation is then sent to the brain and integrated by the brain, and that integration results in the various hues of color, our perception of various hues of color that we have. So we say we have full color vision because we can see across this whole spectrum of colors. And this is enabled in terms of in sunlight by the cones in our retina and by 3 genes that each encode one protein called an opsin. Those are the proteins that are excited by sunlight. So I was asking you, how is it that the kestrel finds that vole? Well, something's happened in the European kestrel. There's been a mutation in the opsin that's most tuned to the blue end, the violet end of the visible spectrum. And that's a mutation, a simple mutation that takes an AGC sequence in the DNA and makes it a TGC, a simple replacement of an A with a T. And that shifts the sensitivity of that opsin into the ultraviolet. Now, I got to tell you, if that happened in you or I, it would really change our perception of the world, right? The violets and the blues would drop out. This would be a unique kind of color blindness in the human population, probably handicap the way we would get around given the significance of blues and purples and everything else in the human world. But what would be bad for us is okay in the kestrel, actually better than okay in the kestrel. And here's why. And this, especially, I know the high school students, you guys are all up there. I want to just show you some of the glories of scientific fieldwork and the kind of things you can discover. And it was discovered that in voles, their urine reflects in the ultraviolet. Okay? Now, I can't tell you how this was exactly collected. I don't know how they could get the voles to pee in the little cups, but that's vole urine illuminated with a UV light, same way you would with a blacklight poster in your room. And so you can see it there, really illuminated. Well, how does this work out? Well, this kestrel can see in the ultraviolet. So that means that the kestrel, instead of trying to spot the animal, simply spots the burrows and the trails, the patterns left by lots of voles going back and forth in the ground, and zeroes in its hunting strategy on those places. Kind of cool, huh? But this is a pretty common story throughout the world, that mutations that would be detrimental to certain creatures are advantageous to others. It all depends upon the conditions. And in this case, if you're a vole-seeking kestrel and you need to tune in on sort of the leavings of voles, and it's actually the voles use their urine as a chemical communication mechanism. So the voles are leaving these things and scents around so the voles can find each other. The kestrel's kind of eavesdropping on their conversations and exploiting that in its hunting strategy. So this innovation of seeing in the ultraviolet is a really simple consequence of mutation. Ordinary mutations that happen every day as any individual kestrel is born, as any individual human is born, with new mutations that didn't previously exist. Okay, so I've told you about icefish and I've told you about kestrels. Well, what about us? Now, the next speaker is going to blow your minds about us. So I'm going to just talk a little bit about what we know, what's going on in the human genome, and give you a little story that's maybe relatable to our deep past, not so much our recent past. So what about the human genome? Well, we know that there's about 20,000 functional genes in the human genome, which is a very similar number to an icefish. A long time ago, maybe only 15 years ago, people thought that humans must have a lot more genes than anything else because we're so complicated and so dang smart. Well, we got the same number of genes as an icefish. Get over it, okay? And in our DNA are hundreds of fossilized genes that no longer work, just like the globin genes of icefish. I want to show you one. It's really my set of favorites, okay? So how do we find these fossilized genes? Well, we've got to find creatures that have sort of the intact version of the gene, and then we can compare them, in this case, with ourselves. So let me tell you about a chicken. Now, some of you guys might have had eggs for breakfast, right? And egg yolks are yellow, okay? And the protein in those egg yolks is made by 3 genes in the chicken. We'll just call them yolk genes. And we know where those yolk genes are in chicken DNA. Now, it turns out, if you look in my DNA or your DNA, in the same corresponding part, because it turns out a lot of genes stay together for a long time in evolution, where the neighboring genes are all the same. And we can find these long stretches in DNA, even at the distance of chicken to human, where that arrangement has been preserved. And when you look in the human, you see all these other surrounding genes, but when you look where the yolk gene is in a chicken, you see just little pieces of code of the yolk genes in a human. Tiny, it's more redacted than any CIA document. It's just got like the and the left. But it's enough for us to tell that there used to be yolk genes there in our DNA. Okay, as far as I know, we no longer lay eggs, okay? So why do we have little vestiges of yolk genes in our DNA? Well, it's because our ancestors did at one time lay eggs, just like that of the chicken. We, mammals, evolved from reptilian ancestors, just as birds evolved from reptilian dinosaurian ancestors. And this history of who we evolved from and the genetic legacy even of the creatures we evolved from is still, over vast periods of time, still accessible in the DNA under sort of good conditions. So that's why you have yolk genes. So if you just, maybe it's table conversation when you guys get home tonight, but talk about the yolk genes in your DNA and see whether or not you can have a little fun with your parents or your siblings. So the interrogation of the DNA record is showing us how variation arises by random mutations. Now, this is a huge aspect of confusion about the evolutionary process. The mutational process is random, in the sense that given 3 billion places in our DNA, mutations pop up pretty much at random over that DNA in every new individual. And that's the same for bacteria, and that's the same for fruit flies, et cetera. And there's no predicting where those mutations are going to pop up. So that's the random process, and that's what Monod meant by the source of all innovation is random mutation. It's like a random number generator. But what happens is ecology. Nature sorts out the winners and losers. The selective conditions, the habitat, cold water, for example, predators, the kestrel, for example, determine which variants are favored, which are the fittest. So mutation is blind and random. Natural selection is not. Natural selection is a competitive process that sorts out the winners and losers depending upon the conditions. So really, when Monod said that chance played this huge role, and he extrapolated from his understanding of DNA and of the genetic code to say that we are the product of an incalculable number of fortuitous events, the result of a huge Monte Carlo game where our number eventually did come out when it might not well have appeared. He really meant, as we now know by interrogating DNA, that thousands and thousands of randomly occurring mutations in our lineage, in our descent from four-legged apes, are what would involved in making essentially the modern human form and modern human physiology. An undirected process, fundamentally a process based on chance. Now, I said he said that with really almost no empirical evidence. And he died in 1976 before we made the discovery, scientists made the discovery of, I think, the finding of the century with respect to the role of chance in life on Earth as well as in human existence. And that's the work by Walter Alvarez and Luis Alvarez and their co-workers that identified an impact of an asteroid or comet 66 million years ago as the cause of the great mass extinction that took out the dinosaurs as well as about 75% of all species on the Earth. This was a radical idea, an idea for which they caught tremendous pushback from even the geological community, that some gigantic catastrophe had rewritten the history of life in just an instant. But now 30 more years of investigation have led to the identification of the impact, understanding of how an asteroid impact can have a massive effect on the climate globally, on the environment globally, and on the extinction of species. So we now understand that about a 6-mile wide asteroid impacted the Yucatan Peninsula, and that shot tremendous amounts of debris out into orbit, which rained back down across the Earth. That hot ejecta triggered wildfires across the planet, wiping out habitats. About 50% of plant species went extinct, so you got to think about how bad were conditions on Earth for plants to go extinct. Tremendous amounts of dust and sulfur in the air that probably blocked out the sunlight for extended period of time, shutting down production across the Earth. It's a really cataclysmic event that's etched and recorded in rocks around the world at hundreds and hundreds of sites that scientists have now studied. But we understand from the nature of asteroids, and there's all sorts of asteroids circulating around our solar system, small ones, big ones. This one just happened 66 million years ago to collide with Earth. And while it took out the dinosaurs, and that was bad luck for them, it turns out that the elimination of the dinosaurs sort of opened up space for small furry creatures around at the time, mammals, including, for example, the earliest primates. And those primate ancestors are, of course, ultimately our ancestors. So it can easily be said that were it not for this one-off event, this asteroid impact on the Earth 66 million years ago, we wouldn't be here. So the role of chance in the course of life on Earth, both at the molecular and the planetary scale, is well established. And this has all unfolded in the last several decades, so I would say, in terms of having a profound insight into the history of life on Earth and the mechanism for generating the diversity of life on Earth, it truly has been the best of times. However, it's not been the best of times for the continuing process of evolution on this planet. I grabbed a headline from just a few days ago from The Telegraph, the British newspaper, The Telegraph. Now, I think you've all read and heard headlines of this nature. Half of the world's animals have disappeared since 1970. Okay, I'd also point out, since we talked about the Beatles a little earlier, that's the same year the Beatles broke up. So it's possible, correlation, not causality, that maybe this is another effect of Yoko Ono. I don't know, okay? So, doesn't seem fair. But to biologists, this is our crisis. We know now, through the historical lens of understanding past mass extinctions, this is a really serious issue. And I want to make really clear, of the large number of biologists that I've been able to talk to about this, the view is we have this problem before climate change enters the picture. This is largely a result of habitat destruction, of over-harvesting, of all sorts of practices of the settlement of the planet by an expanding human population. So climate change certainly isn't going to make this situation better. So much so that you can now go to the bookstores and see several books asking the question, are we headed for the sixth mass extinction? And there'll be TV programs in this realm. And this is not science fiction meant to scare you. There's serious concern in the life science community that the trend line is very worrisome. Half of all animals gone since 1970. Let me give you a telling but worrisome example. Here's a lion I filmed this summer in the Serengeti. Beautiful lioness at sunset. So I was in Serengeti National Park. So African lions, right? There's parks, right? They're protected. Think again. 50 years ago, there were perhaps 400,000 lions across the African continent. If you look in the shading here in orange, this is now their remaining habitat. It's disappeared. Lions have disappeared from 26 countries. And the total population of lions in Africa is now estimated to be about 30,000 from pretty careful counting. 40% of those are in just one country, Tanzania. So here, the king of the beasts, right? It was distributed not only continent-wide, but even into the Indian subcontinent, has in just 50 years, on our watch, been reduced by more than 90%. And you know the same story can be told about sharks and polar bears and other sorts of things. But we also understand from 50 years of ecology that these are not just charismatic animals that we like because we see them in zoos and things like this. These animals play a fundamentally important role in the ecosystems where they're found. And we understand there are sort of domino effects, that when numbers of key species are reduced, other parts of the system suffer. Now, you've heard this message a lot. And the last thing I want to do is just pour on to the hopelessness and despair. Hopelessness and despair are not options. Failure also, though, is not an option, okay? We cannot put our heads in the sand and pretend this is not going on. So I just want to close my talk today by telling you a story from East Africa that perhaps you've not heard. And it takes place in Southeast Africa, in a place that maybe you've not heard of, but I think you're going to be happy to have heard of. And I also forgot to ask the AV folks if they have just 1 second. I hope I'm plugging in sound. If that's going to work. Okay. We're going to find out in a second. Okay, so let me introduce you to a place, Mozambique, which is just southeast of Tanzania. There's a park there called Gorongosa National Park. And it was really well known, for example, in the 1960s. It was one of the destinations for the jet setters of both America and Europe to go there, Hollywood stars, etc. Here's a little footage from that time. All right, Gorongosa, 1963. If you look at the vehicles there, entering Gorongosa National Park, rich in wildlife, a tourist mecca, a wonderful place. But in 1977, civil war broke out in Mozambique and was intense for 15 years. More than 1 million people were killed, and part of the battleground was Gorongosa. And so what happened in Gorongosa in a 15-year strife was that all of the large animals, essentially all of the large animals, were killed. Taken as bushmeat, fed for the soldiers. Villagers had to harvest the wildlife as well to survive. Such that, by the year 2000, if you compare the numbers of various kinds of animals between 1972, before the Civil War, and 2000, after the Civil War, you can see how dramatic the things are here. For buffalo, elephant, hippos, lions, etc. Decimation across the board. But the story's changing. Let me introduce you to Greg Carr. On the left of this picture, an Idaho-born American, a philanthropist and conservationist who formed a partnership with the Mozambique government to try to restore Gorongosa, now at peace, to its original glory. So what's that going to involve? Well, Greg has taken a large portion of his personal fortune made as an entrepreneur and made a 20-year commitment to working in Gorongosa. And he's there. He spends a large amount of his time there. I hope this is going to work. I'm going to let him tell you in his own words what's going on in Gorongosa.
So we're working with the government of Mozambique to...
Do I have sound? Sorry, I should have checked this before.
Greg Carr is committed to conservation here in a way this continent has never seen.
I think we can get, is that live sound right there? Let's try this one again. Let's try this port.
Social infrastructure of...
No? Want to try that one? Tell you what, I'll just tell you what's going on. You want to try that one more time? All right. You may be able to pick up the mic here. It's going to be a little feeble. Let's just run this again.
So we're working with the government of Mozambique to jointly...
U.S. entrepreneur Greg Carr is committed to conservation here in a way this continent has never seen.
I could do the narration of this. Let's not worry about it. All right? I wanted you to hear it straight from Greg. All right, so here's the story. Starting 10 years ago, the comprehensive plan is to restore species, is to even reforest, and most importantly, to make the park part of the community assets, to build schools, to build clinics, and to employ local citizens as rangers, to employ Mozambicans as scientists who are involved in this entire adventure. As I said, it's a very, very long-term effort. So how's it going? Well, they brought elephants back. They brought buffaloes back. They brought waterbuck back. And yes, even lions are back. And now let's look at the numbers 10 years in. A total reversal in the numbers and diversity of wildlife in Gorongosa. The park is there. The ecosystem is potentially there. What we're missing were essentially all the large enough mammals to eat. Okay? So this can be done. And amazingly, the budget for this on American dollar terms is pretty modest. The work is really significant. So now there's all sorts of people from around the world, US academics, Mozambicans, restoring a vast piece of wilderness to the way it looked 50 years ago. It can be done. So I want to give you the example. You're going to hear a lot more about Gorongosa. You're going to see more TV programs. You're going to see more stories about this in the coming years. It's one of the most important conservation projects going on in Africa because it's such a bold reversal of what looked like a pretty much lost situation. So it can be done. So what are people going to say 50 years from now about what's going to happen over the next 5 decades as we face, hopefully, the situation, and we face up to the situation of disappearing animals across the world? Well, most of us have only heard that Dickens quote. They've heard that, the best of times, worst of times. Well, if they made you read Tale of Two Cities in school, I'm looking up there, Dickens, I know I wasn't really a big fan either. But here's what he had to say. So we have to look ahead and we say, is it going to be an age of wisdom or an age of foolishness? Is it going to be the epic of belief or the epic of incredulity? Is it going to be the season of light or a season of darkness? Will it be the spring of hope or the winter of despair? It's certainly true that now we still have everything before us, and in some places we have nothing before us. We're all going to heaven. We're all going direct the other way. This is a choice that is up to us, much as the first 2 speakers emphasized today. These are the huge global challenges that we have to face up to. And I think it's fair to say that we shouldn't be willing to leave the fate of the biosphere to chance. Thank you. Thank you.